Okay, in this video I want to talk about the Watson-Crick model of DNA. Okay, now in 1953, two molecular biologists, James Watson and Francis Crick, successfully described the structure of DNA. Okay, so 1953, James Watson, who was an American molecular biologist, and Francis Crick successfully described DNA structure. Okay. So the same model they described is used today still to describe DNA. Okay, it's known as the Watson-Crick model. Watson-Crick model, basically the double helix model of DNA, right? So that's known as a Watson-Crick model. Here's what it can explain, okay? Here's what the model can explain, okay? So it can explain exact replication of DNA. Replication, duplication, same thing. Okay, it can explain exact replication of DNA. Okay, let me describe this process. So, step one, the strands, okay, are unzipped. So the strands unzipped. I'll draw that. I'll draw an illustration to show you exactly what this means. So the strands is unzipped. Exact copy then made. Okay, by matching each complementary base with its corresponding base, complementary base. Okay, once section is copied, one old and one new zip together to produce duplicate DNA. Okay, so you start off with the strands unzipped. You then get an exact copy made by matching each base with its complementary base, right? So think of hands linking together. And once the section is copied, right, from this to this, one old and one new join together to make a duplicate DNA strand, okay? So here's how it works, okay? So you've got a DNA, right? A piece of DNA. Now let's imagine for a second it's not in a helical structure, right? So let's pretend it's like this for now. Okay, unzipping. So step one means, unzipping means for it to break apart, right? So breaks apart here. Okay, breaks apart. There's some things here. Okay, so this is unzipping. Think of it as a zipper on a jacket, right? Unzips. You then get two new DNA strands, right? So you get this and this, right? Okay, so these two structures here on the DNA that's unzipping correspond to these uprights, whereas these two correspond to these original ones here, right? So I'll repeat that. These two from the base pairs form these two uprights, and the two outer ones are from the original uprights of the DNA, the original DNA. And so then you get the base pairs in between, right? The rungs of the ladder, if you wish. Okay, so what's a mutation? I want to talk about mutations now. So mutation. Okay, it's basically any alteration, any change, any alteration in the structure of genes. And we all know of a very, very common example of this, okay? Spider-Man, right? So Spider-Man has got the genes of a human being. Spider comes along and stings him. One of his genes changes, right? A mutation happens. Eventually, he gets turned into Spider-Man. Okay, so here's an example. So here's a mutation basically in pictures. So let's say I'm going to draw DNA in a slightly different way, okay? Just easier for you to see. Remember ATGC, ACDC, so ATGC, remember? Complementary base pairing. So ATTAGC, GCCGTA. Okay, so here's an original strand of DNA, right? They're the ladder uprights and these are the rungs, yeah, in the middle. Now, if you substitute something, right? If you substitute an extra base pair, right? So AT stays the same, TA stays the same. If you substitute in another TA, right, GC stays the same, dot, dot, dot. So in other words, a single nucleotide replaces another, right? And then it gets deleted somehow. Right, so ATTA is a big gap here, GC, dot, dot, dot. And so it's deleted. So this can disrupt the molecule, okay, by shifting the columns around. So the columns eventually shift around, okay? So if there's a gap, these start to move about. Or if you insert, deleting or inserting, these are two possible ways you can mutate a gene. Or inserting a single base pair can cause a mutation. Can cause a mutation. So that is mutation. Okay. So now I want to talk about So the set of three bases. So the set of three bases, okay, on a DNA code for an amino acid. So amino acids are basically the building blocks of proteins. So a set of three bases on a DNA code for an amino acid and a chain of amino acids are joined together by a peptide bond. Joined together by a peptide bond forms a polypeptide, and I'll talk about that. I'll show you what all this means soon. Okay, poly means many. Okay, it's an acronym that means many. So a polygon has many sides. Okay, many straight sides. So a polypeptide is many peptides. Okay, so and amino acids are the building blocks of proteins, right? So a set of three bases on a DNA code for an amino acid and a chain of amino acids joined together by a peptide bond forms a polypeptide, okay, which forms a protein, which equals a protein. Okay, so back to the Lego that I had, right?